I'm a professor of clinical virology, and I've spent a lot of my life not on autoimmune diseases like Jonathan Edwards, but on viral diseases, and to create diagnostic tools for those. I've also worked a lot on retroviruses, and it was the XMRV story that caught me and got me hooked on the myalgic encephalomyelitis story, because I met a lot of you patients and doctors working with it, and it seemed to be a worthwhile subject for our diagnostic efforts. So I've pursued looking for antibodies in samples from you, and with the idea that this would perhaps give a clue on the reason, the cause of your disease. Of course, this is an assumption, and I will give you some results on how far we got on this. Okay. Well, the techniques that we developed were multiplex tests. So we can measure with these tests about 100 different antibodies at the same time. So we could cover a lot of ground. In a few tests, we could actually test for antibodies to over 900 different antigens from viruses and also bacteria. So I tried to cover those which have been connected with the disease before. For example, the herpes viruses, you know, there are 8 of those. Okay. Now, good. And some of the bacteria which have also been connected with it, mostly intracellular bacteria. And I'll give you a reason for looking specifically at the intracellular bacteria. And that is because mitochondria are essentially intracellular bacteria which were acquired by the eukaryotic cells, the ones that build up our body, 1.5 billion years ago. So the theory was that if you get infected with such a bacterium, you could also not only be immune to the bacterium, but also to your own mitochondria. So that was one of the starting points. Okay, so I'll just talk a little about autoimmunity, because as has been pointed out, it's a big task for our immune system to distinguish self from non-self. And much of this editing of the immune system takes place very early in life, with central deletions in the thymus. But some also must take place later in life, in adulthood, when we encounter different microbes. So that means infections, and that is another tolerance mechanism. But normally this works fine, but you know, it's not easy to distinguish friend from foe. And it depends to some degree upon your some host factors, your immune system, and the recognition of antigens, where you have these HLA, DR2, DR3, and DR4. And some of this was mentioned before, like myasthenia gravis, type 1 diabetes, and so on. So what about ME here? The typical thing for much of ME is that there is an eliciting event, which often is an infection. There are pre-ME situations also, like irritable bowel syndrome and so, which can be there before ME. So the clinical hallmarks, you know all this already, but I will just mention that this is of physical and mental exhaustibility, pain, non-refreshing sleep, malaise, orthostatic hypotension, and irritable gut. That's really a wide panel of symptoms. And then if you try to explain the disease, you would have to find some common mechanism, and that's not so easy. But we've heard a little about what autoimmunity can do. It can really damage both your peripheral nerves and the central nervous system and many other parts of your body. So a clue to what can be expected is to not only look at the ME itself, but the comorbid conditions, which are rather frequent in ME patients, like thyroid abnormalities. Much of those are autoimmune. Orthostatic hypotension, which is recently been found to have autoimmune components, irritable bowel syndrome also, and fibromyalgia. So, as I said, this is how I got interested in this. Another reason is that for suspecting autoimmunity is that many of the autoimmune autoantibodies, they're directed against post-translational modifications, proteins which get modified later on after getting synthesized by adding carbohydrate, or they could be lipids themselves. And many of those are also present in both microbe and human. So that's sort of a central theory for autoimmunity. Another one is that there are cross-reactive conserved microbial proteins, which tend to cross-react also with human proteins. And Professor Edwards mentioned other mechanisms also, but I prefer to make life simpler for me, because I'm not an immunologist. So this is how I see it. So could autoimmunity give rise to the multiplicity of symptoms of ME? So I start with the post-exertional malaise and exhaustibility. So there is evidence that this is caused by deficiencies in aerobic energy metabolism. And so then, of course, I came to think about the mitochondria. And here is a mitochondrion. It's really an energy factory. And, you know, energy from sugars could give rise to energy, in this case, a so-called ATP, adenosine triphosphate, which is the energy currency of our cells. And first of all, glucose, sugar can be degraded to get energy, but later on you can get much more if you get into the mitochondrion. So without the mitochondrion, energy production is anaerobic. With the mitochondrion, it's aerobic. And it's aerobic because in the mitochondrion, oxygen becomes water, and you, in the respiratory chain, gain a lot of energy. That's really a lot of ATP. So if your mitochondria don't work, you will get very much fatigued because you can't use your energy. Mitochondria are, in essence, as I said, alpha proteobacteria co-opted into our cells 1.5 billion years ago. And if they don't function, you get sour. For example, your muscles and your brain will be sour with lactic acid. Okay, there is a disease here called primary biliary cirrhosis, where there actually are antibodies which block the energy chain here at the pyruvate dehydrogenase stage. And so antibodies get into the cell, block here, and you cannot utilize all the energy. So that's actually something that one could use as a model for interpreting antimicrobial antibodies. And there are signs that ME patients have an impaired mitochondrial function. And there is Norman Booth here, who has been working on this. So actually, experimentally, it has been measured that ME patients can have a deficient mitochondrial function. What about the symptoms from the autonomous and central nervous system? You know, there are accepted neurological autoimmune diseases like multiple sclerosis, Guillain-Barré syndrome, and the Tourette syndrome mentioned, narcolepsy, and acute disseminated encephalomyelitis. And in all of those cases, there are more or less clear associations with infections like multiple sclerosis, EBV. I don't think it can explain everything, but it certainly is a statistical correlation. Campylobacter and Guillain-Barré, group A streptococci and the Tourette syndrome, narcolepsy, influenza antigen, and many viral infections can cause the autoimmune encephalomyelitis. So there, as you also heard from Professor Vincent, there are now many examples of post-infectious autoimmunity which confers pathology, especially from the central nervous system. So what about infection here? Respiratory infection by Mycoplasma chlamydia has been connected with ME, gastrointestinal infection, Giardia, mononucleosis, EBV, CMV, or toxoplasma, and protracted fever with neurological symptoms. So all of those in one or other form were used in our antibody screening. But then how can virus, bacteria, and protozoa elicit ME? Okay, so our strategy was to look for antibody rather than the microbe itself, because the microbe might have been there several years ago, but left a mark in the form of antibodies. So it's easier if you have a chronic disease to look for antibodies, although it cannot give the full answers, of course. And this is our technique, suspension multiplex immunoassay. And the antigens were viruses, broad range, and bacteria, and also at least one protozoan. And what did we find here, just to cut it short? A few leads regarding the herpes family, parvoviruses, polyomavirus, and analoviruses, and the chlamydia. So let's see more details. We tested 910 antigens, whole microbe, recombinant proteins, synthetic peptides, with an emphasis on conserved epitopes. Here is one of the most conserved epitopes, which is frequently antigenic, the so-called heat shock protein 60. And it's an enigma how you can be infected with a bacterium. This is a so-called two-dimensional Western blot. And you see here rather strong reactions. This is a rickettsia infection where you get these antibodies. How can one avoid getting pathological effects in a normal infection? So there must be some way of sorting out these antibodies, which cross-reacts also with a human HSP60. HSP60 is a so-called chaperone, which is present in human cells, in mitochondria actually, and in all other microbes except viruses. And this HSP60 binds its substrate through a so-called helix loop I. And you see here the conservation. I'm a little bit short of time, so I will be rapid. You can see that these portions of the alignment are actually both a human and microbial heat shock proteins can be easily aligned. So it's really an ancient and strongly conserved protein. And I direct your attention to this helix loop I. When we tested the blood samples from 69 ME patients and 72 blood donors, first in the so-called discovery set, and then we evaluated the results from that in an evaluation set. We found that there was a tendency to react to whole recombinant protein, HSP60, especially with IgM in the patients, compared to blood donors. It was not a very strong difference, but when we split it up and look at overlapping peptides, small pieces, 30 amino acids, which were across the entire protein, we found that one portion here reacted very strongly and also differentially. The light blue are the patients and the other ones are the controls. You can see it was especially clear for IgM. And that was actually the helix loop I, one of the most conserved and also functionally important parts of the heat shock protein. And if we look at more detail, we see here that it's also strongly discriminating between patients and controls. So we used then the homologs from humans, bacteria, and protozoa. And you see all of them give this difference. So these are highly cross-reactive synthetic peptides, which have a tendency to react more frequently with patients. Okay, was especially clear for the chlamydia pneumonia heat shock protein. Another one that we found that because we ran so many at the same time, we could correlate them. Well, curiously enough, there were also some viral peptides which gave a difference between ME and blood donors. And they correlated with the chlamydia peptide results. And when we do absorption experiments, we see that we can absorb away the reaction to the viral peptide with human homologs, and also you can see with chlamydia. So that there is a cross-reaction between virus and bacterium, which might, please note this is hypothesis, explain why a viral infection could give similar results as a bacterial infection. Okay, so it was mostly this chlamydia and also Staphylococcus aureus, annovirus, parvovirus, and polyomavirus, which gave differences between ME and non-ME. And of these, the chlamydia was the most discriminating. And here you see the results from the evaluation set. Chlamydia peptide from the helix loop I gives a stronger reaction, not at all as big differences as you see with the myasthenia gravis, but statistically significant. So it also differentiates multiple sclerosis, systemic lupus, blood donors from ME. So maybe we have something there, and we hope that this can become a biomarker. Of course, together with the other ones we have. How could these antibodies work? Well, it seems that this helix loop I is really at the heart of the matter for a heat shock protein, and that's exactly where the antibodies bind, and which are then more frequent in ME patients than others. And then we hypothesize that this somehow influences the mitochondrial function. Okay, and another important fact is that this is relatively unique. Other autoantibodies can bind to heat shock protein 60, but they bind in different places. So here is helix loop I, and there are also autoantibodies in type 1 diabetes. They bind over here. Adjuvant arthritis is an artificially induced arthritis. They bind here. And then the chronic vascular disease, where there is a Helicobacter peptide coming from heat shock protein 60. So it's really a protein, one can say, universal autoantigen, is heat shock protein 60, which is not completely understood. But there is a finite and discrete number of autoimmunity pathways which seem to involve HSP60. So the conclusions are that there is an increased frequency of IgM against peptides from this loop, and that there are marked cross-reactions between peptides of humans and microbial and also viral proteins. And the most reactive was a peptide from chlamydia pneumonia, but does not mean that ME is caused by chlamydia pneumonia, because there are so many cross-reactions. Many things remain to study: genetic predispositions. Is there an abnormal reaction in the patients to certain infections? Do the autoantibodies really have a disturbing effect on mitochondrial function? Are there other autoantibodies in the patients? And are there also autoantibodies behind the myalgia? And the comorbidity, I just reiterate that several of them seem to have autoimmune aspects. Okay, so it might be an autoimmune disease. And thank you, too.
Thank you very much. We have time for one or two questions, but I'm just going to take one which someone has written in about. Since we're talking about infection and ME, this question says, can an old virus trigger ME 6 months later? And this is in relation to a patient that is referred to here.
Okay.
I mean, in theory or in practice, I mean, whatever you want.
Well, in theory, yes, because it depends on this interplay between virus and immune system, and that might not take place immediately after the infection. It could take some time. So in principle, yes.
And the other question relates to this infection too, particularly an infection with EBF, but we'll just take it as a virus. Does rest during infection have any effect on the process at all? If you take more rest, would that help with a viral infection and the subsequent effects? Can you reverse any change with a bit of rest?
It wouldn't be bad, I think.
Yes.
But I wouldn't say it's a major factor.
So, I mean, would you encourage people to have rest? Because they say, is it just pointless? They've obviously been told. So, and there's somebody in the audience has asked this question, so they can jump up and say, I'm missing the point if they want, please. But the point is, if you have rest, does it help?
Well, post-exertional malaise is certainly a very important mechanism, and that advocates rest if you have much of the post-exertional malaise. Yes.
Okay, well, the people who asked the question can grab Jonas outside at the coffee break if they want. Yes, sir, we'll ask. Yes, you please.
I've now looked at data of ATP production in mitochondria of over 500 patients. The most distinctive factor is blockage of the translocator protein, the adenosine nucleotide translocator, which takes ADP out of the cytosol into the mitochondria to be recycled into ATP. So more than 90% of the patients have some blockage of this translocator protein. So could that be due to autoantibodies or?
Yeah, the transport of proteins in the mitochondria is governed by chaperones, like heat shock protein 60. So one could hypothesize that somehow interferes with this transport that you talk about.
Okay. Yes, in front. Hi. There's one here, Pierre, down the front.
I have a experience in infectious diseases, and in my career I've seen three very interesting patients, all of whom presented with different infections. One was falciparum malaria, another was chickenpox, and another was miliary tuberculosis. And alongside the classical presentations of those diseases, they all had cerebellar ataxia. And the cerebellar ataxia melted away with resolution of the infections that were present. And I've always assumed that that was a cytokine-mediated effect rather than antibodies, because the way the cerebellar ataxia just disappeared suggested it was a very transient phenomenon. And I have a great interest in ME, and continuing on the theme of infectious diseases, the cognitive disturbance of ME seems to be very, very similar to the sort of cognitive disturbance you find in patients with typhoid fever, for example, and with some presentations of malaria. And I've always assumed, again, that that's cytokine-related and not actually necessarily related to antibody.
Okay. Could I comment then that parasitic infections frequently induce autoantibodies. There are trypanosoma, leishmania, and so on. Sleeping sickness may be due to autoantibodies, actually. So that's really a huge subject which could be investigated more.
So should we be looking for an infection which is a constant source of cytokine in ME? I mean, that would be my hypothesis anyway.
Angela, you have some views on this, I know.
Well, I absolutely agree that cytokines can change brain behavior. That's what malaise is, effectively. It's the cytokines affecting your making you sleepy and making you a bit fuzzy in the head, etc. When you have a flu infection. I think your stories are, like all stories of patients, always very informative. But when you say the ataxia melted away, how quickly? Days, weeks, or months?
Through the patients, the malaria patients.
I mean, that's crucial. You need to know how quickly it goes. If it goes slowly, it's more likely.
The malaria patient came in and was virtually unable to walk. And he walked out of hospital after about 10 days, and there was hardly any cerebellar, there were no cerebellar signs when he was followed up about a month later.
Yeah. Okay, but that's pretty unlikely for an antibody. Yeah. I agree. But in many cases, people say that patients get better remarkably, but actually it took months, and that's much more likely to be an antibody.
I just wonder, could you tell us a little bit about how the intracellular Borrelia causes the dysfunction of the cells? Is there anything known?
Well, I don't know much about that, so why should I say something? Borrelia is a rather hot topic, you know, and there is some overinterpretation regarding Borrelia, as far as I understand, but I'm not an expert on it.
Are there any experts in the audience?
I wouldn't call myself an expert.
Yeah,
However,
Mandy.
there are many theories.
Do you want to get a microphone, Mandy?
I could just be loud. There are several theories as to why there may be some persistent symptoms in individuals, why some individuals with Lyme infection may not recover from antibiotic after an antibiotic course that otherwise seems to be adequate. One is that there may be a host of co-infections. We and other groups are now finding that there are Powassan virus, a whole host of other viruses and other bacteria that may not be responsive to the normal types of antibiotics that we use in Lyme disease. And furthermore, there is another hypothesis that autoantibodies may be induced by either certain Borrelia proteins or other of these co-infectious agents, and that in the long run, what you see for people who are not getting better from the standard antibiotics, it may be that they have an autoimmune process that needs to be addressed.